Right, we're looking at enzymes at the moment, and at the moment we're going to look at enzymes acting as biological catalysts. So, enzymes catalyse metabolic reactions, which are chemical reactions going on inside of your body, and these enzyme actions can be intracellular or extracellular. This means that the enzyme action can happen inside the cells or outside the cells. Enzymes are globular proteins. Enzymes have an active site, and this is where the substrates of the reaction can bind to, and the shape of the active site depends on the enzyme's tertiary structure. So if you look at my other videos to look about the structure of proteins, you can start to understand this a bit more. Enzyme's active site has a complementary shape, so the substrate and the enzyme have to fit together perfectly, otherwise the reaction won't be catalysed, and there's usually only a very small amount of substrates that will fit a certain enzyme. Usually only one substrate will fit a certain enzyme, and as I said, if the substrate doesn't fit into the complementary shape of the active site, then the reaction won't be catalysed, and what the catalyst does is speed up the reactions going on. Enzymes reduce the activation energy, and the activation energy is the amount of energy needed for a chemical reaction to start, and this energy is usually provided in the form of heat. So this means that if enzymes reduce the activation energy, written E with a little a for short, then the reaction can take place at a lower temperature than it could without the enzyme. And the enzyme speeds up the rate of reaction, so it also does that as well as lowers the activation energy usually provided. When a substrate binds to the active site on an enzyme, it forms an enzyme substrate complex, and it's the formation of this enzyme substrate complex that lowers the activation energy. If two substrate molecules need to join together, then attaching themselves to the enzyme holds them close together and reduces any repulsion. In a breakdown reaction, fitting into the active site puts strain on the bonds holding the substrate together, causing the substrate to break up more easily. As you know, only certain substrates will fit in to certain enzymes' active sites because they have a complementary shape. And a way that scientists describe this is the lock and key model. Now, the lock and key model is quite simple. It's basically when the enzyme and a substrate are together in the enzyme substrate complex, they fit together like a key would fit into a lock. So this is the substrate and this is the enzyme, and here's the active site, and they come together to form the enzyme substrate complex, and then they break apart in two, so there's two separate molecules now, and that's called the enzyme product complex, and then the substrate molecules go off and the enzyme stays unchanged. The induced fit model is an improvement upon the lock and key model, and the difference is that there was evidence found that when the enzyme substrate complex was formed, it changed shape slightly so that the substrate locked into place tighter. As you can see here, the enzyme isn't exactly the same, the shape of the enzyme of the active site isn't exactly the same as the substrate shape, but when they come together, the enzyme, the active site, changes shape ever so slightly so that the substrate locks in even tighter. And here obviously it's the enzyme substrate complex, and then when they're made into two separate molecules, it becomes the enzyme product complex, and then they go off separately as two separate molecules, and the enzyme is left unchanged. And this helps to explain why enzymes are so specific and they only allow one particular substrate to bind with them, because the substrate not only has to be the right shape, it also has to make the enzyme change shape so it fits in properly.